Hi, my name is Sabine. I just started my PhD in Neuroscience in the Sjöström lab at the Montréal General Hospital in September 2020, and today I'm going to introduce you to my research topic, the location-specific roles of NMDA receptors in synaptic plasticity. Our brain is a highly dynamic organ, constantly adapting to new demands from our environment. This allows us to solve problems like which bus to take to visit a friend or learning new languages. Interestingly, while the number of neurons is relatively fixed throughout our life, the brain adapts to new information through a change in connectivity between brain cells, or neurons, over time. This process is called synaptic plasticity. Synaptic plasticity effectively alters communication between neurons and, as a result, shapes communication between different brain areas. This underlies virtually all brain function, from brain development to learning and memory, but also contributes to pathologies such as epilepsy, autism, depression, and Alzheimer's disease. Yet, we still do not fully understand how synaptic plasticity is regulated. Now, synapses are the site at which two cells communicate with each other, and usually consist of two parts: a presynaptic cell where the signal originates, and a postsynaptic cell that receives the signal. At chemical synapses, the presynaptic neuron releases molecules called neurotransmitters. The neurotransmitter can then bind to postsynaptic structures called receptors in order to elicit a response in the postsynaptic cell. This way, the signal is propagated, forming circuits and networks. My project focuses on a key protein molecule in synaptic plasticity, the NMDA receptor. The NMDA receptor is a neurotransmitter receptor that plays a major role in deciding which synapses strengthen and which ones weaken with time. In particular, the NMDA receptor can detect when two nearby cells are active at the same time, which is why this molecule is often referred to as a coincidence detector. Coincidence detection forms a key aspect of synaptic plasticity and requires NMDA receptors to be located at a specific site within the synapse. However, this picture is far from complete. Indeed, NMDA receptors can be spotted in different synaptic compartments, suggesting that NMDA receptors are more than coincidence detectors. This is a topic of great debate, and I aim to solve this dispute by revealing how the location of NMDA receptors determines their role in synaptic plasticity. I use a technique called in vitro controllable whole cell patch clamp electrophysiology, which allows me to record electrical activity of 4 individual neurons at the same time at high temporal resolution. This way, I can record from synaptically coupled neurons in mouse visual cortex brain slices, artificially induce synaptic plasticity, and use genetic and pharmacological approaches aimed at NMDA receptors to gain further insight. My project will therefore lay the groundwork for future investigation into how NMDA receptors can be targeted more effectively to combat debilitating neurological and psychiatric disorders such as epilepsy and depression that put a huge personal and economic burden on individuals in Canada and beyond.